Okay, so today then we are going to finish up our biomolecules. Okay, we've been peeling away at this bad boy for what feels like an eternity, so we're going to close this box. So remember, we've been working on our carbs, delicious, delicious carbs, and their variety from simple to complex. We looked at lipids and their variety from food to plasma membranes to steroids. We looked at proteins, right, which had the greatest variety of all, which is why we spent so much time looking at how do we actually fold a protein to get all of that diversity, right? So from primary organization to quaternary organization. So the group work of proteins, right? So we're finally going to land on nucleic acids, which is the last biomolecule that we're going to talk about. And we're not going to spend a whole ton of time actually talking about it, because we're going to spend a ton of time talking about them at the end of the semester when we talk about the functionality and replication of DNA. So this is sort of going to be nucleic acid light, if you will. That being said, when we do this, we're still going to want to do the same thing that we've done for all of our other biomolecules and really focus on what is the point of a nucleic acid, okay, and how can I tell a nucleic acid apart from all of the other biomolecules. Okay. Nucleic acids. All right, so they really have two goals, and that's because ultimately we have two types of nucleic acids, each that have a specialized job inside the cell or body. Okay, the first of which is information housing, okay, which I'm going to call the great library of the cell. The second is the doing of things, okay, the going out and doing something with that information, specifically making proteins. Okay. So we talked about what proteins can do once they're made, okay, and that was what our last mini unit was, right? How do we fold a protein so it works? Okay, but when we're talking about protein manufacturing, we're talking about the actual blueprints for those to even come into existence. Okay, so the step before that. Okay, so the first of these two types is probably the one you're most familiar with. Okay, DNA. Okay, deoxyribonucleic acids. Okay, DNA is our grand library. That's a D. Okay. Because DNA's job is exclusively for information housing, it's big. It's chonky. Right? It's a thick, double-stranded, long, wide piece. Okay? I mean, as far as biomolecules go, it's big. Okay? It's got to be big because it's housing all of this information. Okay, so we really want to imagine this vast library. Okay, like any library. Okay, and inside this library is all of the information that any type of cell could ever need. Right? So if I'm a liver cell, inside my liver, took me a hot minute. Okay, inside of me are all of the instructions for how to make a liver, how to make that liver work and do all of the things that livers fabulously do. Okay, but also is all of the information for how to make eyes, and how to make eyes do what they do, and how to make skin, and how to make skin do what skin do. Okay? This is all of the information ever needed in any cell ever for the body. Okay? Even though it won't necessarily use it, it is there. It's the grand storehouse. It's a lot of information. And it keeps more than one copy of it. So it can never lose it. because it's sort of important information, right? Okay, now it's not going to do anything. This is a storage unit. Okay, just like a library doesn't do anything with that information, right? It is available to you. But the library itself doesn't do anything with those books, right? They're just there, waiting, hoping you will open them. I feel like I'm a dramatic storyteller right now. Open the book. Okay? But they're there, they're waiting, and they're safe. And that is what DNA does. Keeps it safe and stores it until it's ready to be used. Okay, which begs the question, what do we do with it? Okay, well, that's an entirely different job. And that job is the other type of nucleic acid. Okay, ribonucleic acid, RNA. All right, so for as big and chunky and twisted up double time as DNA is, RNA is small and slinky. Okay, because it is not a giant warehouse of information. RNA has a job to do. Okay, so this is like you. When you go into the library, you have a job to do. So you go into that library with a focus. I need a series of books with information to help me write my paper. You are not going to take the entire library out with you. You're just going to take some information with you, right? That information is small and it's targeted and it's going to help you get a specific job done. It's a small amount of information, but it's useful, it's functional, and it's going to go do a thing. And that's what RNA does. It's going to leave the library, go to other parts of the cell, and we're going to talk about in our next unit, tour of the cell, where exactly that is. But it's going to get a job done. Okay, so the DNA is a storehouse. The RNA are the workers that use that information like blueprints to do stuff. Okay, the last thing worth noting is we do have a small amount of diversity in how these things look. Okay, about 5 different types. Okay, not a ton. It's not like amino acids, right? Or we had 20. Okay, but a little bit. With me so far? Not so bad? Okay. So let's look at what nucleic acids actually look like. I was drawing. That was rude. Okay. Everything to the left of my pink line is always the same. Okay, no matter what is going on, no matter which base we have, no matter if we're in DNA or RNA, okay, the left hand of this pink line always looks like this. We always have the yellow bit and the blue bit. Okay, let's focus on our yellow bit. which I'm going to draw in orange because yellow shows up poorly. Okay, we've seen this before, right? These phosphate heads. Why is this so important? Okay, we've got a lot of oxygen, which makes us polar. Which means we'll probably interact with stuff relatively easy. Or at least that part will. Okay? You notice for the blue piece, I say there's 5 carbons. Okay? This is a good way to practice our sneaky chemistry rules, right? So remember, when we're looking at these things, that those bend and twists always have hidden carbons on them, right? We'll look at our bend and twist. So 1, 2, 3, 4, and then we can just see the 5th one. Okay. So this is called a sugar phosphate backbone. And it's called a backbone, right, because it makes a core. Core, it's always the same. Okay. So any diversity we have, all is going to be on the right-hand side then. Exclusively. So those 5 different bases we saw are only going to be different on this last little green nugget over here. Okay, now we see that this is called a nitrogenous base. So nitrogenous literally means nitrogen containing. Okay. Now this is the second time we've seen something that has nitrogen in it. Does that look like what I was doing? Okay. What other group had nitrogen in it? A protein, right? The amino acid base of the protein, right? What did that group look like? What was that called? Close. Really close. Does anybody remember what this was? The amino group. What did it look like? An N with 2 H's. You were so close. All right, so this was our left wing. Okay, so this is the only other time we've seen nitrogen, right? What color haven't I used yet? Oh, I'm running out of colors. Let's go ahead and use yellow. Okay, now the benefit is these actually don't look a ton alike for once. Mercy. Okay. Okay. Our amino group is significantly simpler. Okay, simpler meaning literally there's less to it, not necessarily easier. Correct? Okay. Our nucleic acid looks a bit like Hot Mess Express. Okay. We can easily identify where all the nitrogens are, but this is not a compound I would actually ask you to draw. Okay? Whereas everything else up to this point, I have 100% said you should practice drawing those. Okay? Not necessarily that you're going to have to do it on a test, because I did just say they're multiple choice, but because it is really good practice, and they're not terribly complicated. Right? They're usually 2 or 3 or 4 pieces. This thing, whew, and there's a lot to that. Okay, I struggle to remember that. Okay. So for this, we would just want to remember, okay, it has nitrogen in it. It has more than one. Okay, and all 5 of these have more than one, and there's a lot to it. Okay, that's a pretty complex bad boy. Whereas our amino group, okay, is quite small. And we know that it's 1 nitrogen and 2 hydrogens. Does that make sense? What we would use to compare those two? Okay. What's the other compound we saw a phosphate group in? We've seen that once before, right? Lipids. Good, right? We created phospholipids. Very good. Okay, any questions about anything I have up on the board right now? Or anything about nucleic acids in general? That is it. Told you we were doing nucleic acids light. And this feels real small compared to how much time we spent on proteins. Yeah.
Something like the other ones, you know, just need to know the basic formation for this, the functionality. And you have to know, like, the memorize, like, the G equals C, A equals U. Not that memorized.
No, that's a great question. So she asked if you should have to memorize the base pairing. In other words, that G would be with C and that A would be with T. That is not something you would need to know at this stage. That being said, you will need to know it later on. So it's fine if you start looking at it now, but it's not something that's going to come up right at this minute. Not at this point. Again, this will be something that comes up later, though. Exam 3, I want to say. So a while later. Any other questions? We good online? All right, you guys know the drill. Okay, time for our grand finale. Okay, take out your participation activity, and it's time to finish this beast up. So you're going to add your fourth header. I just coughed into the mic. I'm sorry, guys. Excuse me. I'm gracious. Hey, as always, we're going to have 2 bullet points. Okay, and then the same as always. And we don't have that much to say about these guys, right? So I just need one thing that it was used for. Woo! And one thing that you can use to tell it apart. You do not, no. Okay, as you are finishing up, okay, what you will find on our home page, I'm going to show you in a hot second. I want to make sure I'm not pulling this away on everybody. Is one, make sure that your page is clean and finished for your files, that you have hit save for the final time. Okay. On our home page, what you are going to find, back to home, is where we have these tabs on the left-hand side. If I scroll all the way down, you see that I have a tab for the in-class assignments. When I click on that, I see that I have a box that says in-class homework 1, biomolecules. I assume that's the 9 a.m. class doing that. Inside is a Dropbox. Okay. Those of you that have done this as a Microsoft Word document, you should be able just to submit it as is. Okay. Or you can save it as a PDF and submit it as that. Okay. Be cognizant, Mac and Chrome users, do not submit it as a dot page. It should block you, but just always be very careful. Okay. And if you have done this on paper, also fine, right? What you will need to do is take photos of your work for me, okay? Paste those photos into a Word document, okay, and submit that, okay? Pasting it into a Word document will not only make sure that everything is pasted in as one file, okay, but it'll also let you view your assignment. It'll make sure, one, that you've taken pictures of all your pages so you don't tragically only take pictures of the first page, which happens a lot. And two, it'll make sure that your photos are very clear. Okay, so you haven't stood over them and created shadows or cut off the pages. You always want to make sure that you review your photographs. Because if you've gone to the hard work, you want to make sure that I can see the hard work and I can give you credit for it. Okay. Once you've done that, then just save the file and upload it just like any other assignment. Okay, does that make sense? The Dropbox is open till tomorrow night. So you do not need to rush and get it in right this second. Just ignore it for right now so you aren't distracted during class. There's a regular deadline on there, so it'll pop up nicely for you in your My Courses, like always. But don't forget about it. All right, any questions about anything biomolecule related? All right. Can you stop and restart the recording, please?